Hello, everyone. My name is Xinhai Pan, and I'm a PhD student from the School of Computational Science and Engineering at Georgia Tech. Today, I'm here to present LinRace, which is a single-cell lineage reconstruction method using paired lineage barcode and gene expression data. So first, some background. So I think people are familiar with the single-cell RSG technology. We are able to sequence the cellular transcriptomes of tens of thousands of cells. And combined with the recently emerged CRISPR-Cas9 lineage tracing system, we are able to profile simultaneous lineage barcodes of the same batch of cells. A little more, bit more detail about how to obtain these so-called lineage barcodes. Basically, at the root cell, for example, an embryo cell or stem cell, we inject an exogenous designed sequence, which we call it the barcode, consisting of a sequence of so-called target sites. And we also have this Cas9 protein with guide RNAs, which will induce insertions and deletions onto the target sites of the barcode. In the end, we sequence the barcode of the cells and we compare the mutated barcode with the original reference barcode. We will be able to get the clonal information of the cells. So for the single-cell RNA-seq data, we perform dimensionality reduction and clustering. We will be able to infer the cell types of individual cells. On the other hand, for the lineage reconstruction task, it can be illustrated as following. So on the left-hand side, we have this cell by barcode matrix where different color here represent different mutation states. It can be different unique insertions, deletions. It can also be dropouts. The objective is to infer a tree topology from the barcodes, where on the reconstructed tree, the leaves are the input cells. The goal is to find a tree that is as close to the true lineage tree as possible. And this computational task is extremely challenging, considering the fact that we usually have a very large number of cells. And at the same time, the quality of the barcode data can be very low due to the fact that we have dropouts. So one natural idea is that we can try to use the paired gene expression to help improve the accuracy of the lineage reconstruction. So there are several methods out there. They usually come with the assumption that similar gene expression data correspond to closer distance on the lineage tree. And this assumption is not necessarily true, however. So there are already several papers published to record lineages of various species early embryos, such as zebrafish and mouse. If we overlay the reconstructed lineage tree with the annotated cell types, we are able to get a comprehensive cell fate map and help us understand how the cells divide and differentiate at the same time. If we take a look at the internal nodes, or some people call it the progenitor node, and we look at what is the cell type compositions under these nodes, we usually see that the cell types are kind of diverse, which means that the same cell type can be found in different subtrees. So this observation directly contradicts with some assumption that similar gene expression can directly translate to closer distance on the cell lineage. With this motivation, we really want to demonstrate the complex relationship between the cell types and the cell division tree, and try to improve the cell lineage reconstruction. So we use the concept of asymmetric division, which basically is the case where a cell divides into two cells with two different cellular fates, where one remains at the state of the parent and another differentiate into a future state. So this mechanism is found in stem cells for them to keep self-refreshing and differentiate into functional cell types at the same time. Using this mechanism, we are also able to synthetically generate the case where under the internal nodes, the cell types under the subtree are relatively diverse. So with the asymmetric division model, we developed LinRace, which is a hybrid method using both the gene expression data and the lineage barcode data. An overview of LinRace, we start with the cell by barcode matrix. The first step is to extract the unique barcodes, and we use the classical neighbor joining method to reconstruct the so-called tree backbone. So on this tree, every leaf is a unique barcode, which can consist of multiple cells with the same barcode. Step two is to take the gene expression data, and we run certain trajectory inference method to get the cell state as well as the cell state tree of the gene expression data. For each group of cells with the same barcode, we in step C we will use the maximum likelihood framework using local search and try to find the best tree topology that explains the gene expression data between those cells. In the end, we would attach those gene expression subtrees onto the tree backbone, which yield our final reconstructed tree. I would like to talk a little bit more detail about step B and C. So step B, which is relatively simple. So we have this gene expression data, and we run a trajectory inference method such as slingshot, which will give us a cell state tree. A cell state tree is basically a network that connects the cell types and tells us which cell types can develop into which and which cannot. And in step C, we are taking a group of cells with the same barcode, and they can have different cell types. For example, here, we have a candidate tree. On the leaves, we have cells with the different cell types. And we start with the candidate tree topology, where the ancestral cells states are unknown. We utilize the ancestral inference step to infer the cell state of all ancestral nodes. And then we would calculate a likelihood function based on this candidate tree topology. So for this tree, we propose a novel likelihood function that consists of multiple components. The first one is called the state transition likelihood, which is a joint likelihood of state transitions on all edges. Secondly, we have this asymmetric division likelihood, which is calculated based on all the internal nodes and based on whether or not it divides symmetrically or asymmetrically. Lastly, we have this neighbor distance likelihood, which calculates the likelihood of each pair of leaves being merged as direct neighbors. So these three components kind of evaluates the candidate tree in different perspectives and overall comprehensively evaluate how good a candidate tree explains the observed gene expression data. With that, we would like to test LinRace performances with other lineage reconstruction method, which is relatively difficult because for most of the time, we don't have ground truth for the true lineage. Luckily, we have both like simulated data here, which we know the true lineage, and we also have the true cell state tree. On the other hand, we have this C. elegans L1 larvae dataset where we have the true lineage resolved. Unfortunately, we don't have the lineage barcode data, which will use some simulator to generate the lineage barcode data based on the true lineage. And also, we will infer the cell state tree using the slingshot method from the gene expression data. In the end, we compare the reconstructed tree with the true tree using a metric called RF distance to benchmark these methods. So we selected a few methods, including two methods called Declear and Cassiopeia, which are the some of the top performers in the recent DREAM challenge about lineage reconstruction, which use only the barcode data. And we have a hybrid method called LinkedInMet, which performs maximum likelihood tree search on the whole trees. And we also have two versions of LinRace for simulated datasets. One of them using inferred cell state tree from the gene expressions, another using true cell state tree. From this method, so for RF distance, the lower the RF distance is, it means that the reconstructed tree is closer to the true lineage. We can see that both versions of LinRace outperforms the state-of-the-art methods consistently, which also tells us that LinRace doesn't need a very accurate state tree to perform good, as long as the inferred state tree can capture the relative local connections between the cell states. And we also see that like the other hybrid method performs much worse, probably due to the fact that running local search on whole tree is very inefficient, and they might not have the right assumption to apply the gene expression data. We can also show similar benchmarking results on the real C. elegans dataset here. We also, remember, we generate the barcode data synthetically based on the true lineage. So we test the methods under different mutation rate and with or without dropout effect. And the result seems consistent with the simulated results we shown before. So here we only have LinRace IST, which use the inferred cell state tree. Another finding is that the performance, even though the C. elegans data have a smaller sample size, which only have 363 cells, the performances of all methods seems to be worse than the simulated dataset, which had 1,024 cells. This can tell us that the complicated lineage structure on the real dataset poses more challenge to the reconstruction task. In addition, we demonstrate that LinRace can also provide insights of realistic ancestral state transitions from the reconstructed tree. We use a real dataset of zero-phase juvenile brain that is sequenced using the CRISPR-Cas9-based single-cell RNA-seq experiment. On the left-hand side, we show a LinRace reconstructed tree with the labels that is assigned from the original paper. We can see that the distribution of the cell types are relatively consistent with what we observe from other real data sets. And what we can also do is we use the ancestral inference step in our LinRace framework. We are able to infer the ancestral states for each individual internal node. From the detailed look of the cell divisions of the ancestral cells, we can see exactly when the progenitor cell divides symmetrically to keep self-reflection, for example, the subtree 1 shown, or divide asymmetrically and then develop into a whole subtree of functional cell type, for example, subtree 2 here. With that, I would like to summarize LinRace. So LinRace is a hybrid method that connects the state manifold or the cell state tree and the cell lineage tree using the asymmetric division model. And it's a hybrid method using the joint framework of neighbor joining and maximum likelihood, and is able to outperform the state-of-the-art methods on both simulated and real datasets. In addition, LinRace can also infer the cell states of ancestral cells, which is not permitted for the other methods, and are able to recover more details about cell-fair decisions on the reconstructed lineage. Some of the future work, including basically, I would like to explore more about how the gene expression changes on the distant subtrees for the same cell type, and also how the gene expression changes with different depths, which correspond to different differentiation speed on the reconstructed lineage. And I would like to acknowledge all the co-authors as well as all the other colleagues from our lab. And thank you for listening. I would like to take any questions. I think only tap once.
Oh. Yes, thank you. How much is known about when the cell goes through asymmetric division versus symmetric division? And can you use that knowledge to evaluate your accuracy on your phylogeny?
Yeah, as I said, like validating lineage reconstruction is already a very difficult task. And in order to know when the progenitor cell divides asymmetrically, we need at least time series data where we have data points at multiple snapshots of the gene expressions, and then map the cell types at different stages and find the progenitor cell and determine whether the children cells have different cell types. Which is relatively difficult. Right now, the data set we're working on is only a snapshot of the leaves, and all the progenitor cells are inferred from the data, so we don't actually have the ground truth about that. But it will be a very challenging task in the future.
Can you say something about, like, is your method quantifying sort of the uncertainty of sort of the space, and like how difficult of a problem is it to like return one lineage tree out of like, is it this problem like too unconstrained right now?
Okay, I think, are you talking about the, in terms of their transcriptomics?
Or, I mean, in terms of if the goal is to produce a tree, but then for one tree, but with the input you have, there might be too much uncertainty to actually return one tree, and whether you can quantify that uncertainty.
That's an interesting point. I actually haven't thought about that in terms of how to quantify the uncertainty of different subtrees, determining the self-weight of different subtrees. We use some like probabilistic model in inferring the parameter when we calculate the likelihood, but it's relatively naive, where we basically determine the a certain probability for a cell to divide into a certain self-weight, but which is just a mathematical model for our calculating our likelihood function. But for biology, I think it's much more complex, and we need more experimental evidence, I guess.
Okay, thank you.